My name is Jamie Caroon, and I am the Medical Director at the Center for Medical Cannabis Education in San Diego. Today, I am going to be talking to you about how the endocannabinoid system and the gastrointestinal system talk to one another in both physiological and pathological states. We will talk generally about the endocannabinoid system, what it is, how it works, etc. We'll talk about how cannabis, and more specifically cannabinoids, may help to treat GI symptoms. And then finally, we'll look at one specific condition, inflammatory bowel disease, and evaluate the research that's been published to date on using cannabis to treat IBD.
The endocannabinoid system, or the ECS, is thought to be 600 million years old. It is present in every animal species except for insects, and it is one of the most evolutionarily preserved biological systems that is known. It's fun to think that our bodies produce the same psychotropic compounds that we seek from the cannabis plant when we use it for recreational purposes, but this is not technically correct. We don't actually produce THC or CBD, which are phyto-cannabinoids, but we do produce compounds that are structurally similar and have some similar effects. The compounds that we produce are called endocannabinoids, and endocannabinoids interact with cannabinoid receptors in a similar way that THC and other phytocannabinoids interact with receptors. The endocannabinoid system is an internal regulatory system which functions to restore homeostasis or equilibrium following a cellular stressor. The ECS communicates with all other systems in the body, perhaps most importantly with the central and peripheral nervous systems. The endocannabinoid system has 5 basic components: receptors, the substrate for the endocannabinoids, the enzymes which synthesize endocannabinoids, the endocannabinoids themselves, and the enzymes that hydrolyze or degrade them. Cannabinoid receptors are G-protein coupled receptors. The CB1 receptor is the most densely populated G-protein coupled receptor in the human central nervous system. And there are 2 basic types of cannabinoid receptors, CB1 and CB2. CB1 receptors are most often described as being located in the central and peripheral nervous system. They are depicted in pink in this diagram, while CB2 receptors are mostly located in the periphery, on immune cells and on organs. And this is an oversimplified description of the geographic distribution of these receptors, but we can use it to help explain the model of the endocannabinoid system. It's important to note that CB1 receptors are noticeably absent in the respiratory center in the brainstem, and this is one of the reasons why cannabinoids are so safe, because they do not suppress respiration. And the number one cause of opioid overdose mortality is respiratory suppression mediated via opioid receptors in the respiratory center in the brainstem. People who overdose on opioids basically just stop breathing. Most, but not all, of the effects of endo or phyto or pharmaceutical cannabinoids are mediated through these receptors, and the effects are largely determined by the location and the density of the receptors. The highest densities are found in the cerebellum, the hippocampus, the cerebral cortex, and the amygdaloid nucleus. Receptors in the hypothalamus, for example, are thought to mediate the appetite-stimulating effects of THC, while receptors in the cerebral cortex are thought to mediate the effects of THC on cognitive function. The antiemetic effects, the reduction in nausea and vomiting that are associated with THC, are not as well understood, but they are likely due to activation of CB1 receptors in the vomiting center of the medulla in the brainstem, as well as the hypothalamus and the parasympathetic nervous system. Here we see some of the biochemistry of the endocannabinoid system. Endocannabinoids are derived from phospholipid precursors that compose the lipid bilayer or the cellular membrane of cells, particularly neurons or nerve cells. The entire family of endocannabinoids is called N-acylethanolamines, and they are essentially fatty acid neurotransmitters, which are synthesized on demand by synthetic enzymes. And most of the time you'll hear about anandamide and 2-AG, but there are others. And these enzymes function to convert a phospholipid that is part of the cell membrane into an endocannabinoid. And then there are hydrolytic enzymes that degrade them. And these enzymes basically break them down into their constituent parts and return them to phospholipids. Just a quick point on one of the hydrolytic enzymes here on the left, Pha, fatty acid amide hydrolase. This enzyme is inhibited by CBD, and it has been shown in experimental models that levels of anandamide can be increased by inhibition of this enzyme using CBD. You may have heard the endocannabinoid system referred to as a neuroregulatory system. This is the classic description, and that's the description that we're going to explain in this particular slide. On the left, we have the plant-derived phytocannabinoids, and then the human-produced endogenous cannabinoids. We have a model of neurotransmission here on the right, where we have a presynaptic neuron, the synaptic cleft, and then the postsynaptic neuron here. And in classical neurotransmission, neurotransmitters are synthesized ahead of time. They're stored in vesicles, and when an action potential is propagated down this axon, these vesicles fuse with the cell membrane and spill the contents into the synaptic cleft. And these contents, in this example, are the neurotransmitter glutamate, which is an excitatory neurotransmitter. The neurotransmitter binds to a receptor on the postsynaptic neuron, and a response is induced inside the postsynaptic neuron. Unlike classical neurotransmitters, however, endocannabinoids are produced on demand. They're not stored in vesicles from phospholipid precursors in the cell membrane of the neuron, and they travel in the opposite direction in a retrograde fashion and bind to receptors on the presynaptic neuron, which leads to a reduction in the intracellular concentration of calcium. In this model, endocannabinoids act like a shutoff switch, protecting the postsynaptic neuron from injury due to excessive excitation or excitotoxicity, which is a component of many neurodegenerative diseases. This process is also called depolarization-induced suppression of excitation. And I should point out that this regulatory model is not specific to excitatory neurotransmission. It works the same way for inhibitory neurotransmission, which is mediated by other neurotransmitters like GABA, for example. The endocannabinoid system is more than just a neuroregulatory system, however. It also has autocrine, paracrine, and neuromuscular regulatory functions. This is a diagram of a mouse model of inflammatory bowel disease, where white blood cells like mast cells and macrophages have infiltrated various levels or layers of the bowel. Lipopolysaccharide is introduced in this model to create inflammation and to mimic inflammatory bowel disease. When it binds to receptors on a macrophage, it leads to the production of anandamide and TNF-alpha and other inflammatory cytokines. TNF-alpha is a common inflammatory mediator that is used in studies like this. When anandamide diffuses and binds to receptors on the same cell, the macrophage in this example, this is considered autocrine regulation. When it diffuses to nearby cells like the mast cell and binds to receptors there, that's considered paracrine regulation because it's regulating cells nearby in the vicinity of the cell that produced the anandamide. In both cases, anandamide binding to CB1 or CB2 receptors causes a decrease in the production and secretion of these inflammatory chemicals. These effects ultimately lead to decreased inflammation and infiltration of other white blood cells. From a neuroregulation standpoint, anandamide diffuses to a nearby autonomic neuron of the myenteric plexus, and this plexus largely mediates motility or movement in the gut, as well as intestinal secretions. When anandamide binds to CB1 receptors on these neurons, it inhibits the production and secretion of acetylcholine and tachykinins. Both of these compounds, or these classes of compounds, mediate motility or movement in the gut. And so if their production and secretion is inhibited, then gut motility will be inhibited as well. And so this is the mechanism by which phyto and endo and pharmaceutical cannabinoids may reduce both secretion and motility in the gastrointestinal system.
Peripheral effects are often mediated by cannabinoid receptors outside the central nervous system, like in the enteric nervous system, for example. The three primary plexi which mediate gastrointestinal secretion and motility are the mucosal plexus, the submucosal plexus, and the myenteric plexus. These are all loaded with cannabinoid receptors which mediate the aforementioned functions, as well as sphincter contraction. CB1 receptor agonists have been shown to reduce gastric acid secretion and delay gastric emptying. The same is true for intestinal secretion and intestinal motility. Motility of the GI tract is largely mediated by acetylcholine, and a moment ago we saw how the endocannabinoid system can mediate neuromuscular regulation in the gut by inhibiting the release of acetylcholine. This slide shows some of the different actions of cannabinoids in the gut mediated by these receptors, largely in the three plexi that I mentioned a moment ago. Now we're going to shift gears and talk specifically about cannabis and inflammatory bowel disease, or IBD. I want to present a case for using cannabis as a treatment for IBD. So first, there are anecdotal reports, both historical and more recent, of individuals using cannabis to treat symptoms associated with IBD. And there are studies that show that cannabis use is more frequent in individuals with IBD as compared to those without IBD. There are also surveys of individuals with IBD who report that cannabis is decreasing their symptoms. Now, a survey is a more formal research design than just a smattering of anecdotal reports, but it's still a subjective report by a patient about efficacy, in this case, of cannabis in treating IBD symptoms. These subjects, or these respondents in these surveys, report that as a result of using cannabis and as a result of its efficacy, they're able to use fewer prescription medications to control their IBD. As a result, they have experienced fewer, or are experiencing fewer, adverse effects, and they're experiencing a greater quality of life. There are also preclinical studies in both human and rodent models which suggest that the endocannabinoid system is involved in the IBD disease process, which we just saw in the previous slide, and that cannabinoids may have therapeutic effects. And then finally, there are some clinical studies involving humans that indicate that perhaps cannabinoids are reasonable, safe, and effective treatments. So let's look at some of these a little bit more closely. Studies in mice and rats with experimentally induced colitis have revealed some interesting findings about the role of the endocannabinoid system in IBD. First, CBR knockout mice, that is, mice without cannabinoid receptors, have a higher risk of colitis than mice with cannabinoid receptors. And this is true, frankly, for a variety of inflammatory and autoimmune disorders. Secondly, Fa inhibitors, that is, agents that block the enzyme fatty acid amide hydrolase, which is the enzyme that hydrolyzes anandamide. The inhibitors of this enzyme increase anandamide levels. And by the way, CBD does this as well. CBD has been shown to inhibit this enzyme, which results in higher anandamide levels. In addition, these studies have correlated those higher anandamide levels as a result of inhibition with improvements in GI function and inflammation in rat models of colitis. And then finally, we have experimental models. THC and CBD have been shown in these models to reduce the severity of tissue injury, to reduce inflammation, and to reduce the production of free radicals. So we see both endogenous and exogenous cannabinoids are having multiple therapeutic effects in these experimental models. Moving on to humans, there are studies which examine intestinal biopsies of humans with IBD, and these are ex vivo studies. They reveal, among other things, decreased levels of anandamide in these biopsy specimens, decreased expression of the enzymes that synthesize anandamide, increased expression of the enzymes that hydrolyze anandamide and other endocannabinoids, like Pha, as we just mentioned. And they also show an increased expression of CB1 receptors in the biopsy. And you see this in a lot of different tissue types, including cancer biopsies as well. Clearly, the endocannabinoid system is disrupted in some way during the disease process of IBD. Whether this is a cause or an effect really remains to be seen. Moving up the evidence-based pyramid, a case study was published in the Annals of Internal Medicine in 1990 that described a 23-year-old female who used cannabis to successfully maintain remission of her ulcerative colitis. In 2011, a retrospective study was published which included 30 patients with Crohn's disease in Israel. These patients had disease that was refractory to steroids, immunomodulators, and anti-TNF alpha agents. The investigators asked the subjects retrospectively about their use of cannabis and the corresponding effect on their symptoms. All 30 patients or subjects rated their general well-being as improved, and 21 of them reported notable improvements in their Crohn's disease symptoms with cannabis use. The average Harvey-Bradshaw index for all patients improved from 14 to 4.7, and that was a statistically significant finding. The Harvey-Bradshaw index is similar to the Crohn's disease activity index, which is basically a composite measure for measuring the severity of the activity of the disease at a given point in time. Also of particular interest in this particular study was this idea that a number of patients were able to either discontinue their steroid or reduce the dose of it. So the number that required steroid treatment was reduced from 26 to 4. The following year, a prospective observational study was published with a single arm. It included 13 subjects with long-standing IBD, refractory to conventional treatment. These subjects were given 50 grams worth of pre-rolled joints and told to smoke them whenever they needed to. And at the end of the study, they asked them, and that was 3 months, a number of different questions. They generally reported improvements in daily activities, improvements in quality of life. They gained weight, they gained body mass index, and again, the Harvey-Bradshaw index reduced from 11.4 to 2.9, also statistically significant. The next year, in 2013, a randomized control trial with placebo was published. In this study, 21 subjects with Crohn's disease activity index scores above 200 whose disease was refractory to standard care were randomized to either receive pre-rolled joints or cannabis cigarettes, which contained 115 milligrams of THC twice daily, or placebo cigarettes. The primary outcome of the study was the induction of remission, which was defined as a CDAI score of 150 or less after 8 weeks of treatment. A secondary outcome was what they termed a clinical response, which was defined as a decrease in the CDAI of more than 100 points. This is figure 1 from the study. You can see the two groups compared by their Crohn's disease activity index scores at 3 time points: baseline, week 2, and week 8 of the study, and then at one follow-up visit at week 10 after treatment. Notice the decrease in the scores in the treatment group from week 0 to week 2. The effect was fairly immediate, and then the increase after cessation of cannabis use after week 8. You can see that the scores basically returned to the baseline after treatment. 5 out of 11 of the subjects in the cannabis group experienced complete remission, which was the primary outcome, while only 1 in 10 in the placebo group experienced it. This finding was not statistically significant, but that is probably due to the small sample size in this trial. So to summarize, we have seen patients and subjects report positive, disease-specific outcomes in anecdotal reports and in surveys. We have seen retrospective and prospective observational studies where smoked cannabis provided clinically significant symptom relief based on the Harvey-Bradshaw Index and other outcomes. And then we saw a randomized controlled trial with a placebo that showed clinically significant but not statistically significant symptom relief based on the Crohn's disease activity index. Only one study, the RCT, told us anything about dose: 115 milligrams of THC in a 0.5 gram cigarette smoked twice daily.
Thanks for your time and attention. Please reach out to me if you have any questions.